All right, welcome back. So what we're going to take a look at now is some of the formed elements of the blood. So well, generally they call them formed elements. I mean, in reality, they're mainly cells. That being said, platelets technically are not cells. They are actually fragments of cells. So calling them actual cells is a little bit incorrect. So what we're going to see is there's three main formed elements and then some subtypes within certain levels of this. So you have erythrocytes, which are also known as red blood cells, platelets or thrombocytes, which are pieces of a big cell called, megakaryocyte, there we go. And then finally you have leukocytes, of which there's two main types of these, but these are white blood cells. They have ones that have cytoplasmic granules that you can see, which are called granulocytes. These are neutrophils, eosinophils, and basophils. And then you have the ones that you do not have visible granules in the cytoplasm. Those are the agranulocytes, which are the lymphocytes, and the monocytes. The overall process of this takes place in the bone marrow and is called hemo or hematopoiesis. All of our blood cells actually start from these blood stem cells called hemocytoblasts, and then they can differentiate into all the different types of blood cells. Like I said, there are two main lines that they can produce. And again, I'm not worried that you know the whole differentiation process here. I want you to know that they all start from these hemocytoblasts. And it's really, you get this myeloid cell line that is going to be your erythrocytes and everything except for the lymphocytes. And you then have the lymphoid cell line, which is the lymphocytes specifically. These are regulated by different growth factors. So you can get things like multiple colony stimulating growth factor, granulocyte macrophage colony stimulating growth factor. The presence of these different growth factors are what are going to trigger this to differentiate in a particular direction. I'm not going to ask you how and which ones are triggered which ways, but I just want you to see that there is this kind of high level of growth factor regulation that is going to determine this differentiation pathway. So you can see there's a couple other stimulating factors here. There is also something called thrombopoietin, which is going to lead to the production of platelets, as well as erythropoietin, which we'll talk about honestly in a lot more detail a little bit later on, that is involved in the formation of red blood cells and increasing the rate of production of those. If we talk specifically about red blood cell production, that is erythropoiesis. And again, these are the vast majority of the formed elements. Over 99% of the cells are erythrocytes. We're going to see that it does require certain things like iron and vitamins, certain vitamins and amino acids for these to be produced. But they are honestly what's being made more often than anything else. Leukopoiesis refers to production of the leukocytes, both the granulocytes and agranulocytes. And again, the granulate cell line is coming from the granulocyte cell line of the eosinophils, basophils, neutrophils, coming from that myeloid stem line and differentiating those granulocytes, where the monocyte cell line is really just going to be those monocytes that become macrophages. And then finally you have the lymphocyte cell line, which is from that lymphoid branch of this that are going to either become B or T lymphocytes. And we're going to come back and talk about those a lot more detail in the immune system and what is going on with these different types of lymphocytes. Thrombopoiesis is the production of platelets. We're going to see you get this fusion of a bunch of cells called a megakaryoblast, and then it becomes a megakaryocyte, and it's little pieces of this that are going to break off and become these proplatelets, which sooner or later become these platelets. And this is just kind of showing you the old branching here. Again, I want you to know the hemocytoblasts and really the finished cells here. I'm not going to ask you to explain the differentiation of a basophil versus a lymphocyte. I just was trying to point out with all the stuff here that the process is tightly regulated and it is controlled a number of different ways with different hormones, and make it clear that this is tightly regulated. Again, there to me is enough things to talk about and enough things that you need to know that I'm not going to walk you through all the little pieces of this and make you know every step. But again, know that they all start from that hemocytoblast and that we have two main lines, that myeloid and lymphoid line, and then really know the finished cells. So the erythrocytes, platelets, and the different leukocytes. The only other one I would say is know what a megakaryocyte is. So that's what I would like you to know, at least in terms of this differentiation process. If you were to take this and make a blood smear, do the staining, this is kind of a small example of what it's going to look like. You can see all the different formed elements. Again, in reality, you're going to see a lot more red blood cells and platelets than you would see white blood cells. So this is not really realistic in terms of a real image. You would generally see maybe one leukocyte and all red blood cells and some platelets mixed in. But this is showing you each of these different leukocytes and what they kind of look like. Again, we are going to come back in detail and go through each one, what their job is and how to identify them. But to start with, I want to kind of begin with your erythrocytes. So these are the red blood cells. They have a particular shape that is very distinct to them. So this is a biconcave disc. I would say kind of like a donut or a tire without the center part being completely gone. This does a couple different things. First off, it gives us a lot of surface area. So the main job of these is to carry oxygen, and to do this, they want more surface area to have that oxygen to be able to get in there. So this biconcave disc is really going to decrease the amount of volume relative to the surface area, which allows these to take in oxygen much quicker. The other thing that this shape does is it allows it to be a little bit more flexible. So you're going to see certain capillaries are only about 1 cell width thick. Because of this, these cells, if you get a big circle and the hole's a little bit smaller in a circle, there's no way to have that fold up to get through there. It's just going to bulge and block. So by having this biconcave disc, it means they have some flexibility to kind of bend one way or the other, and that flexibility allows them to get through these smallest vessels. And again, these are generally the smallest full-size cell that's in here, about 7 to 8 micrometers across, very consistent size. And really, when you look at this, the cell is really a cell filled with the protein called hemoglobin, which is going to carry oxygen and carbon dioxide. You notice a couple of things about the erythrocyte that really makes it a lot different from other cells. One of the things that is very obvious when you look at it under the microscope is it does not have a nucleus. So what does no nucleus mean for us? One of the things it's going to mean is no nucleus means the genetic material is not going to get copied. You're not going to get cell division of these cells. So these are a cell that is not going to divide and make more of itself. These are fully differentiated. They don't divide. It's those stem cells that are making large numbers of these. So these cells, because of that, are going to have, and the other thing is no nucleus means you can't make proteins and other stuff like that, which means these cells are going to have a limited shelf life. So I always kind of say these end up becoming disposable cells. They're made, they last a certain amount of time, they wear out, are destroyed, and that's really what goes on with them. So the other thing we're going to see is no mitochondria. Why is that an important thing? Well, mitochondria uses oxygen to generate energy. This cell is not doing a lot of metabolism, first off. The other thing is, and it's kind of a bad analogy, but a drug dealer doesn't want to be a user of his own stash, what he's supposed to sell. And it's kind of the same idea with erythrocytes. You don't want the erythrocytes using up the oxygen they're supposed to be carrying. That would be like a pizza man that is always just kind of taking the pizza and eats the pizza and never delivers it. So by the fact that they don't have this mitochondria means they can't really consume oxygen in order, it means they're actually good carriers of oxygen and can transport it around. So hemoglobin is this important protein in there. It is actually one of those proteins that has that quaternary level of structure. So it is made of 4 chains, 2 of this beta type and 2 of this alpha type are these heme groups. And then within each of these globin chains, you're going to have these heme groups in the center, which at the center of each of those heme groups is going to be an important molecule, important element. So if we look at these heme groups, this is the structure of it. And again, I'm not so worried that you know the structure, but the center molecule, this Fe, that actually stands for iron. That is the periodic table for iron. This is the active site of each heme group. At that iron, you can bind an oxygen molecule. So per molecule of hemoglobin, you can bind 4 molecules of oxygen. And oxygen binds to hemoglobin in a fairly weak fashion. So each molecule, like I said, can bind 4 molecules, but it's fairly weak. And you might kind of go, well, that doesn't make a lot of sense, so you want it to carry it. Again, we want it to carry it, but we also want it to deliver it. And again, it's like You don't want a pizza man that is so into pizza that he will eat everybody else's pizza and never drop the pizza off. So we want this to be like a pizza man that is kind of sick of pizza, so he just wants to always deliver the pizza. So that fairly weak attachment means it will bond, but it also can get delivered and released to the tissues. Carbon dioxide also binds fairly weakly to this. We're going to see carbon dioxide is carried in multiple ways within the blood, but one of the ways is on the globin portion of the hemoglobin, and it actually helps with the oxygen unloading at the tissues. One of the things that is kind of interesting, and it can show you what happens when you get something that binds too tightly to the hemoglobin molecule, carbon monoxide, so that is CO, not CO2 like carbon dioxide, but carbon monoxide, has a affinity about 200 times higher than oxygen. So what tends to happen with this one is if you get exposed to carbon monoxide at high enough levels, it will preferentially bind to the hemoglobin groups and outcompete oxygen, which means you cannot pick up oxygen through your hemoglobin, and all of a sudden you cannot get enough oxygen and you will lose consciousness, and if it's high enough, you'll die from it. One of the ways they do this, they put you under pressure to try to get more oxygen into you in this hyperbaric chamber. Sometimes if you've had too much carbon monoxide poisoning, even that won't do it, and really the only way to treat it is to do a blood transfusion with blood that does not have any carbon monoxide bound to it. We do give some different names to the hemoglobin depending on what's bound to it. So you have oxyhemoglobin when it's bound to oxygen, deoxyhemoglobin when it's free of oxygen. And then if we have carbon dioxide bound to the globin portion, we call it carbaminohemoglobin, which I just think is fun to say. And really, the function of the erythrocytes is there to really transport this oxygen to the tissues, pick up carbon dioxide, take this back. So those are the main things here. They does also provide a buffering system for the blood in terms of blood pH, partly because of how we carry carbon dioxide is going to influence blood pH. So that kind of gives you the idea, the basics to start on these erythrocytes. What we're going to spend some other time doing now is they can look at some of the blood types and what that means to us in terms of the erythrocytes, and that's what we'll do. So I will see you next time.